So I'll just introduce our discussion moderator for today. So Dr. Bassett, who leads the Cellular and Gene Editing Research Group at the Wellcome Sanger Institute, where his group works as part of the Open Targets Consortium to dissect the causes of neurodegenerative disease.
Brilliant. Thanks very much. It's a pleasure to be here. And yeah, once again, I think please do put questions into the Slido. But while you're all doing that, I might start with one or two. So I thought that was a great talk. First of all, I think it's and the proposal is fantastic. I think this sort of ability to couple genetic variation to kind of phenotype in a completely agnostic way, I think, is very, very powerful. So one thing that sort of sprung to mind, I mean, so you mentioned multiple different cell types. So how important do you think the cell types are, and sort of, and stimulation conditions within those cell types as well?
Do you want to take
I can take. I mean, we, as Melina mentioned, ideally we would love to run every single cell type that you can, but one needs to be really quite practical about this approach. We would see the benchmarking phase to be very critical and informative for the scale-up and for the mapping phases. And in the benchmarking, we should be ambitious. We should think about a large number of cell types, but equally be practical, because you can think about taking a cell type and running it through, let's say, 10 stimulations, and the space very quickly grows. So our initial proposal was between 8 to 10 cells as a practical, feasible approach that would equally be informative for the next phases of the project, prioritizing on the disease relevance and diseases where there's strong genetics, so that we could really make the largest impact, as well as diseases that perhaps are of unmet clinical needs, where therapeutic interventions are really necessary.
Right.
Great.
And maybe just one adding point, this was perfectly capturing also my opinion. Just to add, one of the considerations in collaboration with the New York Stem Cell Center would really be to think about which differentiation protocols are robust enough to not, Andrew, we had it earlier, to not be heavily confounded by the differentiation variability of iPSC models. Yeah,
absolutely. And in terms of sort of how you balance that with stimulation as well, I mean, so there's obviously, you've got a certain number of cells that you can actually interrogate. So you've shown very clearly that sort of the stimulation and the time courses are very, very critical. So how do you view that kind of balance? What's the kind of...
We start out really broad in this kind of benchmarking phase with only 10 individuals, so you can span your entire phenotype by state matrix kind of over different time points of stimulations, differentiation in the respective cell type, and then use these data to subset and select the stimulatory conditions and stimulations really that appear to have a maximum effect.
Okay,
there's also one other note which would be beautifully integrated with BodyMap, and truly in the last executive committee meeting we discussed this, would be to take from the tissue level, single cell level profiling, and then really use that to ask how much of the cell populations do we recapitulate in the dish with a certain differentiation protocol. Is that reflective of disease biology or not, right? So there's great, complementary.
And I guess, in immune diseases, we have the benefit that often we know, for example, which cytokines are at elevated levels or which are successful drug targets. So, you could take that as an additional opportunity, include those, and then look where you really see major differences. And I think the benchmarking phase will be very revealing in terms of identifying where you have redundancies or certain, lack of responses, or responses that are very specific to a cell type and time point, in which case you may not maybe want to take that into the next phase and much rather focus on a stimulus that has a much broader impact.
Yeah. Okay. Cool. Great. Okay. So another kind of aspect to it, I guess, is kind of, you were talking about the genetic diversity in UK Biobank. So, and Melina, you also very nicely presented the kind of extremes of PRS scores. So how do you kind of view sort of the genetic diversity and how that kind of sort of adds into this? Or where do you get it from? And what sort of genetic diversity would you have?
Yeah, but that's very, very, very important for sure. This is actually, in terms of the biobanks we would be tapping into, a major consideration to be really reflective of ancestral diversity. For the early UK Biobank discussions that Ben, Cecilia, and we had, we hear that there might be a way of upsampling certain ancestries when prospectively recruiting patients. But also we, see this, you said this one so nicely, the open call to the community of biobanks that really want to engage. We've heard Andres and today talking, I don't know where he is, prospective ancestral biobanks will really be wonderful to pull into this effort, right?
Yeah.
So obviously UK Biobank is the biobank sort of in the community, but equally there are smaller biobanks, yet they have very rich phenotypic information, and in some instances they may have complementary information. So just an example, coming back to Carl, there could be biobanks that have very deep microbiome phenotyping, for example, that could be relevant to IBD, and it would be worth seeing if there are opportunities for partnering up. So whilst we're super enthusiastic to have UK Biobank support, we'd love to see if there's an enthusiasm from other biobanks to join forces on this.
Cool, great. Carl.
Yeah, I have a question around that number of 10,000 individuals. I don't need to justify that versus 15,000. Don't worry, that's not my question. How you then partition that number of experiments across cells, stimulations, phenotypes, and if we take that kind of old rule of thumb, that the closer your phenotype is to the nucleus, the fewer individuals you need to map genetic variants associated with it. I mean, cellular phenotypes, I guess, are further away from the nucleus than expression or kind of molecular regulatory traits, but they're a lot closer, I would imagine, than common complex diseases and common complex traits. So have you thought then kind of what your minimum unit size is in terms of number of individuals? I mean, if I was plucking a number out of thin air somewhat, I'd be thinking somewhere between 200 to 300 individuals, and then be thinking about that as my unit and going across several traits and several cellular phenotypes. Is that what you're thinking of, how to partition across these multiple different things you might want to maximize across?
Yeah, so, I mean, very much. So, I could guess maybe around 200. I know of one study that looked at the effects of genetic variants on T cell proliferation, and with 250 individuals identify 1 or 2 hits. So I think we don't really have much data to infer these numbers, and that's where the scale up of 1,000 individuals will be hugely informative. But I would suggest that it will be more than 200 probably to be well powered to detect effects on cellular phenotypes, and I would suspect more at the order of 500 to 1,000. We'd love to see this as an ambitious project, and 10,000 also is partly capturing the genetic diversity, right? If you go on with 2,000 individuals and you try to split that across different ancestries, you become quite underpowered within each individual ancestry.
Yep,
Is there anything?
absolutely. Agree. Power calculations we have started discussing, and I look to Ben, which as I recall from discussions with Ben and Cecilia, you were also in line with the kind of 200 individual mode of starting to map. Power calculations for cellular phenotypes are really difficult. Exactly, nothing has been. It hasn't been mapped so far. We have no idea, but. I think it's a confident number that gets us into the space of.
Ben and Claire and I actually did preliminary power calculations a while back, and roughly 400 cell lines in duplicates should capture the most sort of low-hanging fruit variants that we have seen. If you look at the reported V to F sort of individual loci follow-up data and the type of effect sizes that people report in those papers. Now we know that's probably a gross overestimation of the effect sizes you're likely to see. So extrapolating from that, and that there is only really a handful of robust signals out in the literature, has led to that calculation. But calculations are apparently happening somewhere in Ben's group.
Just very, sorry. I can just very quickly add, actually, there is a paper on the cytokine responses in PBMCs, and they had about 500 individuals, and there were 17 genome-wide significant hits. So which kind of agrees with this calculation as well, that 400 to 500 individuals should give you some.
The more, the better.
What do you see the value of protein interaction maps for assessing cell function and its perturbation by mutations?
Very high value, though. So as I mentioned, or did you want to? The selection of, so we discussed many different assays that we would be pulling into such a CellMapper tool. The problem with protein-protein interactions is that it's not scalable to this scale, right? And also very difficult to optimize for individual cell systems. I don't know if Casper is here. No, I don't think so. Who we are working with very closely in terms of protein-protein interactions. What I do believe, though, is that for a subset of individuals, protein-protein interactions can serve as anchors for the interpretation of protein networks across differentiation trajectories or stimulation responses, etc. So yes, feasibility consideration is we couldn't pull it into a CellMapper tool across all individuals, but I think it's definitely a very valid additional phenotypic modality to read out.
So it's a challenge.
The scale is a challenge.
Scalable and technical.
Correct, yeah.
Anything else in the room? Maybe I could ask another one. So this is maybe David as well as Melina, but in terms of the sort of phenotyping, I was quite struck by the fact that with the sort of staining techniques that you were doing, you could actually pull out understandable features.
Right.
And I was wondering sort of what your thoughts were on kind of the number of features and how important that is for interpretation later on.
So in our experience, the number, in terms of feature space, the signature matters, right? What I have shown you are features that map to the, granularity features, intensity features, location-based features at the edge of the cell, which really make a phenotype that then can link to insulin resistance. Mitochondrial intensity itself as a feature would have not given us that cellular program. So I do think, granular extracted features are important. It comes, of course, with the downfall of multiple testing burden. So it really is a balance to take there. And we typically, per channel, we are working with 700 features per individual marker that we are mapping onto relevant traits. Interpretation, of course, and Dave, you can chime in here. Interpretation is a very it's a challenge in terms of high content imaging. I can talk on behalf of my lab. We are working on this for 6 years now, and the majority of time has been going into his computational analysis and interpretation of the actual features and making sense out of them in terms of pathways. Such a multimodal data set is incredibly informative of individual features as well.
Yeah, that's a good question. I personally like to view it as the, what's the actual information content of the staining that you're doing. Some staining methods, like cell painting, like afford the opportunity to extract, large numbers of features. If you're doing, say, like a live cell imaging assay, and you've got like a couple like fluorescent reporters that are reporting on a signaling pathway, obviously they're going to be like much less, and typically the features that you extract for them to be useful have to be done in a very particular way. The interpretability is, as Melina noted, is a very big deal. You could actually categorize a large part of my lab's work under that umbrella of interpretability, because the single cell lens is, one of the lenses, that humans use to make these sorts of data more interpretable. Yeah, so I guess the answer or non-answer I would give is it depends.
There's another question over here.
So something that I really like about this approach, this PRS-based approach, is that it's not contingent upon identifying causal variants and causal genes before moving on to identifying the cellular processes. I think that's a real strength because, as we heard earlier, it's so hard to identify causal variants in genes right now. On the other hand, it might be valuable to know the variants and the genes in particular that are underlying the processes you identify, and probably, if you're looking at PRS, it's not going to be the case that all of the variants in the PRS are affecting whatever process you identify. So I guess my question is, to what extent do you think it's going to be valuable to go back after identifying processes to the genome to identify the genes, and how do you think, what types of approaches are going to be needed in order to do that?
I think highly valuable, absolutely. Like kind of the way I sketched this was as an acceleration of the dropping the identification of the causal variant scheme. I think for the CellMap approach, this type of data set, the systematic data set, should inform and prioritize and help find mapping efforts to narrow the regions of associations down to smaller windows, right? So I think, this is, as Gosia mentioned, in terms of developing statistical frameworks, computational strategies, I think that's a sandbox to really play in, and I do think it's very important to kind of afterwards go in and ask, are there certain variants with a higher probability? Rare variants, when we go into the 10,000 donor space, would be particularly valuable. Kind of this gene dosage curve that I highlighted was getting a little bit into this to establish causality of a certain pathway, right? Yeah, I don't know if others have additional thoughts. Is that capturing it?
Yeah. I guess followed
Luke,
on from that,
is there any?
Sorry.
No, I completely agree, especially about rare variants and identifying rare variants that act on some of these same processes as the common variants. I completely agree.
Right,
Yeah.
I guess following on from that, do you think there's any scope in adding some engineered variants in there to actually act as anchors, if you like, to sort of, so if you knock out a gene or you sort of upregulate a gene, that might give you an anchor to compare back other sort of variants to?
That would be the perfect comprehensive project, and I think what the CellMap team would have loved to do is pitch both. Though that's not feasible to tackle from the get-go. But I completely agree, Andrew. This is, this kind of natural genetic variation mapping of cellular processes, and then the validation through high-throughput pooled CRISPR perturbation screens. We work with JT Neal on in situ sequencing, as I mentioned. I'm currently setting up the technology for pooled CRISPR screens with high-dimensional imaging readouts. This is, yeah, absolutely, prospectively.
Right.
I was Jesse Ingrites from Stanford University. I was just going to, I think, make a similar point, which is once the benchmarking phase is done and one has identified the time points and stimuli and cell types to focus on, I think it would be feasible to do, genome-wide CRISPR knockout screens, for example, on those same conditions, and have, Luke, to your point, to have a map of the genes that control each of those cellular processes as well.
Absolutely, yeah.
Great. Okay. Is there any other burning questions? I notice we're up to the hour on time, so might call it a day there, unless there's anything else. Great. Thanks again to all the speakers and the panelists, and yeah. Great discussion.
So thank you, everyone. This concludes our plenary session for today. Just to remind you all that we have a reception to refresh you all at 5:30, and then followed by a dinner at 6:30 that I'm sure everyone will be well ready for by then. We'll start day 2 tomorrow at 8:45. So we'll see you bright and early for another day of exciting scientific talks. Tomorrow we'll focus on body map and disease map working groups.
Bravo.
Oh, and there's a satellite meeting for the Bravo team as well. Okay. Thank you, everybody. Thanks so much.